this is a project that is done by Adam Gilewski, and who's a master student of this project, and myself. And initially, we also had help by Barbara Hawkins, who was at this meeting too, and Samantha Robbins. They are the experts on measuring, doing the so-called electrolyte leakage assays that we are doing. Barbara is also a co-supervisor of Adam on his master's project. Yeah, so we're looking for identification of natural yellow cedar variants in above average levels of root freezing tolerance. And we already talked, or Vanessa talked, give a good background to the risk factors for that. And just wanted to touch on that, why forest trees are so vulnerable to rapid climate change. And it's quite obvious, they can't move. They have sometimes very long generation times, which is slow compared to the changes that we're seeing in many ways. And they have limited seed production, especially yellow cedar has fairly low seed production and fairly low spread then over time. So they don't move and they don't spread fast. An example of that is aspen dieback that you may have heard of, not just in Canada, but in the US too. This is an old map, so it doesn't show the whole extent of it. There's quite a bit more in Manitoba than is shown here. So you can see that you have aspen parkland in both Alberta and Saskatchewan that is quite extensive and has suffered quite a bit of aspen dieback, and there is more of that further east, as well as in western USA, higher elevation then. And this has been linked to drought. Of risk factors are not fully understood because there's a disease that comes with it, it seems like. And aspens are poorly prepared in the sense that they tend to form large clones. They propagate vegetatively by shoots from roots, and you get very large clones. So that means that we have limited genetic variation to respond to these changes. An example closer home is yellow cedar decline. This is a picture from Paul Hannon, and here's a map that I think Vanessa showed, the same map, but about 200. These are already old numbers now from 2016, over 200,000 hectares in Alaska, and more than 50,000 hectares in British Columbia that are affected by yellow cedar decline. And Vanessa also talked about the risk factors, and the most obvious one, which is the environment where you find yellow cedar, is our wet soils. When you have wet soils, both because they're sitting on a boggy lands, or they're sitting on mountain sides with a lot of melting water, they develop shallow root systems. So that's one major risk factor. The other risk factors too, like open canopy conditions that generates exposure, and exposure to cold snaps in the late spring, and then of course you have soil warming in early spring, especially so with global warming, but also reduced snowpack or no snowpack, and these are risk factors that now have been linked to freezing injury in this tree stand and yellow cedar decline. And here's a picture of a simple experiment that shows how this can, well, it can be mimicked, the lack of snow then. So these are potted seedlings grown in Alaska at low altitude, where there's no snowpack anymore in the early spring or late winter. And these ones have been, instead of snow, then have vermiculite on top of the soil surface, whereas these plants do not. And you can see the plants without vermiculite die, whereas those that are covered with vermiculite do not die. And there's a good agreement between low snow accumulation and yet the cedar decline particularly in Alaska, where it has been studied well. But this is not the only risk factor too. So Vanessa has looked extensively at Haida-Gwaii and the decline that happens there too, also with historically no snowpack in much of the areas. So shallow roots, is that really true? It's hard to believe for many people that they would have so shallow roots that they would be exposed to freezing damage from cold snaps. And if you look at boggy sites, the trees and the bushes, yellow cedars are there. They have at least when I've been looking at them, very shallow roots, but with most of the roots just a few centimeters below the ground. And then they often grow at mountain sides too. When you have a landslide like this, you realize that how limited the soil depth is at both sides. So what can be done to mitigate effects of well, yellow cedar decline? Well, if it's to be done, when an obvious solution would be assisted migration of other species to fill the yellow cedar niche. But sometimes the other species cannot really grow on both sides. They're simply too wet and too poor for other species. So if it could be possible, it would be assisted migration of yellow cedar decline resistant yellow cedar. And then the big question is, can we identify yellow cedar decline resistant yellow cedar? So these are the hypotheses that we have for Adam's project, that natural variants with relatively high freezing resistance do exist. And also that survivors in yellow cedar decline areas may be more freezing resistant. So you have a survival of the fittest natural screens going on in those areas for freezing resistant ones. But also that high altitude yellow cedars may be more freezing resistant, not because they're not, they do have snow coverage, but perhaps they have a higher ability to resist lower temperatures because of a harsher climate. But it could also be that yellow cedars further south may be adapted to no snow and early de-hardening relative to those further north. So we don't really know. So these are working hypotheses. And the long-term hypothesis then that natural freezing resistant variants could be used for reforestation to mitigate the spread of yellow cedar decline. And maybe, if not directly, that they also could be used in a breeding program to select for offspring that have superior resistance. So what we have done is, this is a small project of the past 2 years funded by BC Parks to collect foliage in yellow cedar decline areas, but also high altitude and further south. Bring the foliage to the lab to root cuttings and grow roots, cold harden the plants, and test relative freezing tolerance of those roots. And so rather than screening in nature, so to say, bring it to the lab and use highly standardized conditions. And we're still learning how to standardize those conditions to be able to pick apart a relatively small difference in freezing tolerance. And the first trip was, I was really lucky, very thankful for getting on a helicopter ride with Stefan Seglen and Sari Sanders, who's going to talk soon, up in the mountains close to Prince Rupert. You can see Prince Rupert Airport is out here, and here's a work channel, and the helicopter took us to a few sites, not so high, up to 250 meter heights perhaps, but still huge difference because it's very hard to get to those sites without a helicopter, or it will take a full day to just get to one site. So we saw extensive evidence of old and extensive yellow cedar decline, like these old spears here, but also trees that are just now dying. And here's Stefan. Well, you see just his bum here, but he's standing here and looking at this tree. He's the expert on looking at these trees and see what's going on then. And I ran around and I collected not actually so much foliage for cuttings, but we're not good at rooting cuttings at that time. So I collected branches that had rooted naturally by layering. You can see a few of them here. And then I brought them to the lab and tried to keep them alive and grow roots. Later on, I went back to the same area. It was a canceled boat trip with a ranger due to bad weather. So I rented a car and drove around to a few sites. So this is just east of Prince Rupert, along the highway here. Diana Lake Provincial Park here is a nice site. And here you can see what has been described as fairly typical symptoms of yellow cedar trees that are dying due to yellow cedar decline then. So we actually start to make these clusters of extra branches and before they eventually die then. So these are not dead trees, but highly abnormal trees here. So I collected both from proper trees like this, but also from bushes at wetlands at a few sites along the highway here, Gamble Creek Ecological Reserve, Diane Lake Provincial Park, but also the road that leads up to the Works Channel and other sites. On another trip, Adam and I went with a ranger in the Douglas Channel. So you can see that Prince Rupert is up here. This is Terrace over here, Kitimat over here, and this is the Douglas Channel here. And we were basing this collection trip on two things. First, the maps like this that shows in green the spread of yellow cedar as described by BC government maps, but also airplane trips that had mapped cedar decline. So we were looking for areas of cedar decline that had been recorded close to the coast of this channel here. But it turns out that we really had a hard time to find any yellow cedar trees. We found a few sites, like for example at a distance, this one here that we think is yellow cedar decline, but it's pretty high up on the mountain slope. And in another place is Hartley Bay out here, where we could really walk up to them and see extensive yellow cedar decline, both small and big trees and long dead trees, as well as more recently damaged trees. So we found very few trees. We traveled for 3 days with the ranger on a boat here to various declined, and so few trees. And another thing that we noticed is that the seedlings are almost completely absent. We found on all these trims, we found a grand total of 3 seedlings. Lots of hemlock seedlings and so on, but even under western red cedar, yellow cedar trees, no yellow cedar seedlings. And I mentioned before that that it didn't seem to agree with maps we had on yellow cedar and what we found. And this is the government map on yellow cedar spread on the coast. It's range, and this is a blow up on this area with Prince Rupert here, Terrace, and the Douglas Channel here. And on the trips here, both as well as several days on traveling with a car and hiking, all these yellow crosses means that we couldn't find any yellow cedars. And this is not just low altitude, but also higher altitude, like for example, the skiing area up here, going all the way up to the top and spending a whole day looking for yellow cedars and not finding them. So the places where we found them are quite limited. The green dots here, primarily close to Prince Rupert. I'm sure we can find them at higher altitude, but it seems to me that these maps are actually grossly overestimate yellow cedar distribution based on what we can see here. And on top of that, areas that have been marked as yellow cedar decline by an airplane, actually what we found when we went to them with the boat is that these were actually not yellow cedar trees. These were either western red cedar trees or alder and perhaps also cottonwood trees that were dead. So due to COVID, we couldn't travel in the north the following summer. So we traveled to Provincial Park in the south. But we focused then on, and that's where you will find yellow cedars, that they grow at a higher altitude and not as low altitude as further north. So we were at 800 meters altitude and above, and we focused on mountains where you can actually get up with a road first. So skiing mountains, Cypress Mountain, Mount Seymour, but also Pinecone Burke and Hemlock Valley, which is out in Harrison's Mills. And we collected trees that have different exposures, those that well developed and very large trees. This is my son cutting a branch here with a canopy, but also trees that are, this is Cypress Mountains that are on top of one of the peaks here with almost no soil. So these trees would be exposed to, especially if there's no snow coverage, then would be very exposed to cold snaps. Here's another example. This is from Spipeus Provincial Park on the Sunshine Coast. This is actually an area that is not supposed to have yellow cedar, and I didn't know that, which was lucky because we found plenty of yellow cedars up on these mountains. So you have Madeira Park here as a reference on the Sunshine Coast. Here's another one on the Sunshine Coast, fairly close close to Seashelt, the Tetrahedron Provincial Park. And there I saw plenty of signs of yellow cedar decline. So you have the old spears here in a fairly wet area, a little bit lower altitude. But here at a high altitude, I couldn't get to this site, so I can't say for sure that this is yellow cedar decline or even yellow cedar. But if it was true, then that would suggest that this would be a further expansion south of yellow cedar decline than that you could see as an arrow here, quite a bit further south and has been recorded, at least to my knowledge, of yellow cedar decline. So this is just a summary of South Coast collection site, except for the Hemlock Valley, and we focus then on higher altitudes. So we have altitudes from 800 meters up to over 1,200 meters altitude collected here, around 200 individuals. And then we have learned to route by trial and error, I would say, cuttings from adult foliage. That's what we're doing in these tents. We build two tents. This is an old picture. We have 2 tents with misting. So we cut off cuttings and put them in rooting hormone and a fairly loose peat vermiculite mix and have them misted a number of times every day. And eventually the root takes up to half a year before many of them actually root, and many of them also come to rot. So this is a numbers game. We have to put a lot of cuttings in here to get some rooted. And this shows some of those rooted seedlings from different clones. This is one tree, one branch that resulted in this cuttings. There's another branch that resulted in this cuttings, and so on it goes. And now we have a few benches, I think something like 500 to 600 individuals growing right now. And then I'm going to leave it over to Adam, who talks about how to test the cold hardness.
Yeah, hi. So we take those rooted cuttings, and what we do to kind of measure freezing tolerance between individual genotypes is use a electrolyte leakage assay. So the sort of theoretical basis behind it is when you freeze the root tissue, ice crystals start to form, which damage the cell membranes, and then electrolytes can leak outside of the cell membranes, which we can use as a proxy for freezing tolerance. So the more leakage there is, the less tolerant the individual is. Next slide, please. Oh, yeah. So just to go over how the assay actually works, we take root tissue from these trees, we chop it up into smaller segments and put them in a glass vial. We then introduce a few ice chips just to initiate ice nucleation, and we gradually freeze those tissues. We also include unfrozen controls, which are just kept on ice during the duration of that experiment. Distilled water is then added, and the vials are shaken overnight to allow the electrolytes to distribute into the solution. The next day, we can measure the conductivity of the solution. So the voltage changes based on the amount of electrolytes that have leaked into the water. Those samples are then heat killed and measured again, and those measurements can be used to calculate an index of injury, which is roughly equal to a percentage of the total available electrolytes that have leaked due to freezing damage. Next slide, please. So this is just an initial experiment I did with trees from a singular mother plant, you could say, so from one branch, Diane Lake 1. And here you can see we cold hardened some trees by keeping them in a cold room for 2 months with short daylight conditions. You can see that the hardened trees have a lower index of injury than those that have been unhardened at these various temperatures. And because the temperature of -4 was closest to about 50%, we used that for further experiments. So these are results for a few genotypes, but these are from those rooted branches that Jim was talking about. So I could only get technical replicates from these. So technical replicates, I had just one plant, but then I took roots from that plant and distributed it among multiple glass vials to kind of get technical replicates. You can see that there is some significant variation between these. These are just preliminary results that likely have confounding factors that have to be confirmed. You can see the ones on the mostly to the left, those are ones from the helicopter trip. So you can see within those areas, there may be some variation. These middle ones, AL6, I just included as a sort of control between weeks where the experiment was run separately to see how much variation there is by using technical replicates. And those are from Stecklings specifically. And then the GC and the GT are from the road trip. Some of these are going to be cut out by Zoom, unfortunately, on the far right. But there's also variation between Gamble Creek individuals, which suggests there may be variation within the same kind of general regions. But yeah, these definitely need to be repeated with Stecklings to kind of confirm these results. And then I also did the experiment with some Stecklings that we did have at the time. For the most part, these are all from the general Prince Rupert area as well at lower elevations. As you can see, they're mostly around the same amount of index of injury. One individual, Work Channel Road 4, had significantly less leakage than these 3 other individuals marked in blue. One of them also being from Work Channel Road. Oh, I should also mention that the Work Channel Road 4 individual only had technical replicates, so that's definitely something that should be repeated as well. And then when I just look at the 3 highest and lowest leakage rooted cuttings, you can see that AL3 and AL03-7 may also have potentially lower leakage than other individuals. But we may want to look for individuals from different altitudes and latitudes that may have more significant differences than these ones.
Yeah, so this is Jim again. So I'm just going to summarize briefly what we have done so far. It's still early days. We collected from more than 300 trees in northern and southern BC from I would say close to 0 meter altitude to 1250 meter altitude. We have rooted branches and cuttings, and many of them are still coming through. So that's why we haven't tested that many yet. We have so far screened only 29 lines, I should say, from the Prince Rupert area, 0 to 200 meter altitude. But in such a small set, we actually see differences in freezing tolerance already now. So we're fairly optimistic, something that we didn't know if we would find at all. And we do seem to see differences in freezing tolerance between some of these lines. And we are also going back to this big trees that we collected in the beginning with helicopter. We have cloned them, made many clones, so we can test them. Can we repeat this with clone material and reduce variation? And we're starting to screening high altitude trees in January. So, and I want to just thank some people that helped us, Stefan and Sari, for the helicopter trip. It was a wonderful event. We also went with Ranger Stefan Ben, that helped us to get in the Douglas Channel, and GitGat First Nation too at Hartley Bay, that provided some trees as well as housing for us when we were visiting there. This is what we see. We also saw some humpback whales. Adam had some mishaps with jumping off a boat into deep water and things like that. But otherwise, very interesting experiences. So thank you so much for your time.